Hello everyone, my name is Ditya and my project revolved around studying the neuroscience behind itch and inflammation, specifically how the absence of GFR-alpha-3 affects the conditions of atopic dermatitis. Now for a little bit of background, atopic dermatitis is a chronic inflammatory skin disease that currently has no cure, and nor does it have any adequate treatment options. It also affects millions of people around the world. AD has been linked to diverse triggers in the past, but the exact neural mechanism behind it remains poorly understood. However, there have been reports saying that artemin plays a significant role in itch and inflammation. Artemin is a neurotrophic factor that binds to its protein receptor GFR-alpha-3, and the dorsal root ganglia, or the DRG, is responsible for transmitting sensory signals from the peripheral nervous system, or the skin, to the central nervous system, or the brain. Now, if we take a closer look, there are populations of GFR-alpha-3 and TRPV1 on these nerve endings, and that is how GFR-alpha-3 binds to artemin, which is located in the skin. Now, the reason we were investigating GFR-alpha-3 was because it binds to artemin and was thus predicted to also play a role in itch and inflammation. Now, with that being said, the central purpose of my project was to investigate what would happen if GFR-alpha-3 was eliminated from the neural circuit. I had two primary objectives. One, to semi-quantitatively recognize differences in various mRNA like TSLP and artemin in AD by isolating RNA and conducting qPCR to see how these gene expression levels might change in an AD victim. Secondly, I was also meant to analyze the effects of MC903 on GFR-alpha-3 wild-type and knockout mice by conducting behavior analysis after the full MC903 treatment. Speaking of MC903, this mechanism is a common tool used in AD research to induce atopic dermatitis. The mechanism is known to increase epidermal thickness before and after treatment. And this figure shows that by day 10, the mice did in fact itch more than when compared to the vehicle-treated ones. Now, I was primarily interested in inflammation and itch. For the inflammation, again, I isolated RNA from euthanized MC903-treated mice skin tissue, made cDNA, and conducted quantitative PCR to amplify our genes of interest. For the itch side of things, after the MC903 was applied, the mice were recorded on day 10, and I conducted behavior analysis. The behavior analysis looked something like this. The mice were recorded in IITC containers, and I kept track of how many itching bouts occurred every 5 minutes up until the 1 hour mark. Now, this is an image of a mouse during an itching bout as its hind paw is scratching the back of its neck. As for my results, the first experiment I conducted was to show that MC903, when treated with it versus the ethanol, which was our control, there is upregulation of TSLP and artemin in the skin. Now, while IL-17A did not show this change, that was somewhat expected because it is a marker for an entirely different skin disease. However, since we just established that TSLP and artemin do increase under AD conditions, we expected that knocking out GFR-alpha-3 would cause these expression levels to go down. Interestingly enough, however, we saw that there was no change, as shown in figures 11 and 13. As for itch behavior and skin thickness, a reminder that day 10 behavior showed that the mice did significantly itch more and their skin thickness significantly increased by day 10. However, again, we saw no difference in these two results. Now, what are the major takeaways? There was no significant difference in itch behavior and skin thickness progression between the two genotypes. There was also no difference in TSLP and artemin expression between the two genotypes, even though we showed that MC903 implementation did cause upregulation in TSLP and artemin. Now, while we did see GFR-alpha-3 did not have an impact on itch behavior or skin thickness, it might be time to try the artemin ligand itself. A way to do this is through artemin sequestration, which is effectively a way of neutralizing and debilitating artemin from later activating GFR-alpha-3. Now, we saw with the pain model that previous results showed that artemin sequestration did block hypersensitivity, but GFR-alpha-3 did not. Therefore, it might potentially work in the itch model as well. As for the neural circuit, trying to knock out other components of it might also help us understand their roles better in atopic dermatitis. In the neural circuit behind AD, there are various neuropeptides and ligand-gated ion channels. So like GFR-alpha-3, if we knock out other ones, we might have a better chance at understanding the neuroscience behind the entire thing. With every step we take, we come closer to discovering more and reaching our goal in the field. Thank you, and a special thank you to the NCSM Foundation, Burroughs Wellcome Fund, NCSU, and the Mishra Lab. And thank you for your time.